Ready? For drugs. Okay. We're going to do a lot of drugs today. You won't believe it. So we're going to erase this. So we're going to get a little ahead of what we cover in lecture, but nothing that when lecture comes will be easy. But we're going to make a little neural network here that's involved in something called the reward pathway. So we have a pathway in our brain which is, gives us pleasure and reward, goes to various centers. And the idea of that is we have natural things, but that's where a lot of drugs, that are legal and illegal sort of affect that reward pathway. And we're going to do something to show you the neural network. In that sense, I'm going to draw 3 basic neurons. Or 4 actually. And this will be reward. Reward, pleasure, good feelings. Okay? Now the idea of this is that now we're going to deal with a couple things. First of all, we're going to deal with a neurotransmitter. There's a major neurotransmitter that's involved in the pleasure system, which is dopamine. Okay? This central guy is basically going to be the neuron that if we go through the action potential, what's at the end here is going to be releasing dopamine. So again, just to make it sort of simple here, this is a dopamine producing neuron. If it fires, we didn't quite get to the end. We got all the way to here. What we're going to see next time is if the action potential reaches the end here, we're going to then release the neurotransmitter, and it's going to affect the next cell. That's the end of that chapter. But here we're just going to say, okay, if it fires, it's going to release dopamine. If dopamine is released, it's going to act upon this postsynaptic cell and fire, come to the reward center, basically, again, it's several parts of the brain, but create the feelings of pleasure, reward, etc. It's how much dopamine is going to be related to how much reward. Now, the idea here is I started to indicate, and we'll see next time for sure, that when we're talking about signals coming in to a neuron, whether it's going to fire or not, some are excitatory signals, which means closer to threshold, more likely to fire. Some are going to be inhibitory, which means we're going to make it more negative, less likely to fire. And there's not 2, there's hundreds coming in, we'll see. It's always going to be a balance between positive and negative. If positive reaches enough, which will end up being enough so that we have threshold here, we'll fire and release dopamine. But if we don't have enough, or we have a lot more negative, this won't fire at all. Okay, we won't reach threshold, no action potential, no dopamine released. So that's more like a real neural network here. We're simplifying it and making like one negative guy coming in and one positive, but there's hundreds of these. So we step back. Here's a picture of the reward pathway. Do we basically get it? Are there any questions right now? Because that's sort of the basic of starting. Okay? Now, two things we have to know. The video is going to help us. But when we're talking about controlling neurotransmitter, this will be another slide coming up, like we release it. Okay, there's 2 ways that we can sort of control it. Because if we have dopamine here, and it's going to affect the cell, do we want it just to stay there and keep affecting the cell? And, well, we might, I guess. But in general, let's say in general, for neurotransmitters, we want some sort of control here, right? Now, there's 2 methods of control that the video is going to talk about that and the lecture, but I want to reintroduce it. So one way we control it is called reuptake. Kind of using, and you'll see this video, it's very visual, reuptake pumps. And that's actually what we'll see how we control dopamine. So dopamine will be released, but then some of it will be constantly taken back up in the cell. That controls how much dopamine is active in the synapse. Okay? That's how we normally control dopamine, basically. Something's causing it to be released, but we're also reuptaking it to be used again, and that way, we're controlling the level of dopamine. Okay? And that's one way. Another way is going to be something called, we're going to basically break it apart using enzymes. So it's enzymatic degradation. We're going to see here in the video, and I'm going to talk about, we're going to be releasing a neurotransmitter called acetylcholine. Summarize ACH, acetylcholine. We're going to see, and I'm going to actually make kind of columns here with the video, that actually is going to be controlled by enzymatic degradation. They're going to make an enzyme that chops it up, and that's going to control it. We're releasing it, it's affecting the cell, but we're also chopping it up until we're at the right balance. And those are the two ways that we sort of control the amount of neurotransmitter. And that's going to be important because guess what the drugs are going to do? Mess with that. Okay? And again, it really doesn't matter whether, in my mind, whether we're talking prescription drugs or we're talking illicit drugs, they're all affecting sort of the levels of these neurotransmitters. And that's what we're going to show. Okay, now we're ready to show. And before the video, though, I'm going to drop the chalk once more. Because I love talking. We're going to fill this board up with drugs, okay? You're going to write this down. It's good stuff. And so what I want to write down, what I'm going to describe is sort of the effects of, on the system, the effects of, here, cocaine, the effects of nicotine, and you want to throw some heroin and opiates in? Sure. Heroin, various opiates. The video won't actually go into heroin. It will talk nicotine, cocaine, and everybody's favorite?
Ecstasy.
THC. The THC. Don't worry, we'll get to ecstasy. So the video will talk about those three. Okay, and we're going to revisit this as far as that. Now, the last aspects, we're going to fill this board up like I say. The first part of the video, and again, some of the questions on that sheet that we're going to end up answering, specifically, it's good because it kind of talks how these neurons generally work, and it's going to be saying the differences between two neurotransmitters that you're going to want to know. And that's going to be, I'm going to put that here, I guess. And we're going to make sure that we know these answers. So even if you don't quite catch it on the video is interesting. But it's going to describe the differences between how acetylcholine works as a neurotransmitter and how dopamine works. Okay? Okay, that's the first half of the video. The video is like 25 minutes. And the guy with the video, it's very visual, but it's got this kind of druggy music, and the guy sort of talks weird. And you're going to see he's got bad drugs. Because the second half, he talks about, again, THC, nicotine, and cocaine, and really crazy things are happening when he's on those drugs. Okay, so, I'm not saying, these drugs are okay, don't worry about that, but there's some crazy stuff on that. Okay, but that's the second half. Okay, the last two drugs, I'm just going to tell you how they work, okay, after this video. Just because I started to add it. And how many people watch Breaking Bad? Isn't that a great show? So I just did this last week. I had to look up how that methamphetamine works. Okay, so meth. It's such a good show. It's not even funny. Okay, so in fact, on the PowerPoint for these other classes too, I got a picture. Both of them up in their suits, like making it, and So we're going to show you exactly what it does, okay, based upon everything we're learning. And then let's throw in a little LSD, okay. See, that's back in my day, okay. Okay, LSD, and then of course, I don't know if I spell it right. I don't know this drug at all.
It's a C instead of a.
What is it?
An
Wait, say it again. E what?
ECS.
Ecstasy. East.
No, we know what you mean. We know what you mean. And then it's a C and a C and X instead of S.
Okay, you remember those dendritic spines? They're gone. Okay, a little too much. Okay. long time ago. Okay, so here's our drug mixture, okay? This is what we're going to learn today, how this actually works. Just in time for the weekend. All right. Let me take a drink. This is much more fun than like membrane potential. All right. So we're gonna again. I want a different piece of chalk. Where cocaine is going to work is here. And so I'm going to bullet point it. What cocaine does is block dopamine reuptake. And you'll visually see this in the video. So I'm just putting dopamine reuptake. Okay? Cocaine is working here. Now, if you block the reuptake of dopamine, what does that do to dopamine levels here?
It spikes it.
And that means what's going to do the reward? That's what we're doing. Blocks dopamine reuptake. That means the dopamine's going to stay there longer. Okay? Greater dopamine in the synapse. It's not releasing more. It's just that it's going to, whatever got released is going to stay there longer and affect that cell. And that does mean happy. Greater reward. That's how it works. Okay? Simple as that. Next. Okay, so we did a little cocaine, but let's throw a little nicotine in there. Now, nicotine is going to work here. And the video will show this. But what's happening with this guy called positive, it normally releases acetylcholine that binds to its receptor and causes the nerve to fire. It's positive, right? Nicotine basically acts like a acetylcholine agonist. What does an agonist do? It does the same thing. And so it's going to bind to that receptor, and basically there's a lot more nicotine than there was. This release causes this to be hyperactive. And so when that happens, let's just follow this now, and I'm going to bullet point it. So nicotine, which is an acetylcholine agonist, will bind and activate this, which means it's going to release more dopamine. And more dopamine means more reward. So nicotine basically binds to the ACH, acetylcholine receptor, I'll put activates, but the idea is it's going to basically cause the neuron to fire, activates this dopamine neuron to release more dopamine. That's a difference. We're actually releasing more dopamine. And more dopamine means more reward. So, whereas cocaine just kept whatever was there longer, nicotine is actually going to stimulate this neuron to actually release more dopamine. And now more dopamine, greater reward. So a completely different mechanism. The end result, which is what the pleasure pathway cares about, is dopamine. Two different ways to do it. We're going to go to a third way in a minute. So one way cocaine blocks reuptake, that dopamine stays there. Nicotine actually will stimulate this, causing more release of dopamine. You ready for a little heroin? Little heroin. Now, is Jesse going to find out that Walt basically killed his girlfriend? Okay? That's what I wonder. Because if he finds that out, that might be the end of it. Anyway. What? Did what? Did I see Sundays? Are you kidding me?
Oh my god, he might not even find
Oh, they're not going to kill Jesse. If they killed Jesse, there would be like this massive, revolt. They're not going to kill Jesse. You're going to get out of it somehow, okay? There's no doubt about it. But it was so good he got it right. It's like, I'm burning your money. And it's like, oh, the guy went freaked out. Oh, it's such a good show. All right. Sorry. But we're not doing that yet. We're doing heroin first. That just reminded me of his girlfriend. I liked her too. She was like cool until she was on heroin. All right. So we go to this. The idea of this is, and this is sort of an important concept, because if you want to increase something, if you want something to fire, let's just put it over here. If you want to, and then I'll erase it. If you want to, increase activity or firing of a neuron, there are two ways you do it. You increase the positive, and/or what do you want to do to the negative? Exactly. You inhibit these inhibitory. Let me actually just write that. Inhibit the inhibitory. What's negative times negative?
Positive.
Positive. If you look over here, because again, this is the framework of this. Okay, one way we stimulated this, the nicotine, is what? We made the positive greater, because this came in. And again, by positive, we're not doing charge. We're talking about something that's basically causing it more likely to fire, right? We're causing more of a depolarization. But here's the negative influence. If we decrease that, who's going to win the battle?
Positive.
Positive. And that's the two ways. If we're doing it, we're always doing both of them, right? If we want something to fire, we'll increase the positive signals and we'll decrease the negative. So we want to turn it, sort of in the language we're using, we want to turn this neuron off so it's not releasing the negative. And then this is going to start winning. Guess what heroin does? Okay? Turns the negative off. So here in the other opiates, binds to this guy. Okay? If you turn this off, it's not sending inhibitory signals. Positive's winning, and when positive winning, more dopamine. More dopamine means more reward. So, heroin opiates inhibit the inhibitory. It's that double negative stuff. Inhibit the inhibitory neuron. Okay? That means, basically, what happened here. That means because negative's less, positive's going to win, so we're going to have more dopamine release, because this is going to fire more often now. Okay? We got rid of the negative, this fires more often. Again, more dopamine release. I know that was... I'm getting all excited on drugs. More dopamine released. And more dopamine released means what?
More reward.
That's exactly it. So what's nice about this system here, and we're going to see the video now before we do the other drugs, and we're going to fill this in, make sure you get it. What's nice about this system here is, all three of these increase dopamine and reward, but by three different mechanisms. So you could combine them, and you really will get a lot of dopamine and kind of a lot of reward. There might be a couple side effects like death. Okay, but cocaine, again, just to summarize these three, and then we're going to see the video. Cocaine creates a system higher because it prevents reuptake of dopamine, so dopamine stays there longer, a greater effect. The nicotine stimulates this neuron, acts as the natural neurotransmitter, so it's an acetylcholine agonist, stimulates this neuron to produce and release more dopamine. More dopamine released, greater effect. Heroin works over here, shuts off or decreases the inhibitory signal, so this positive signal, whatever it is going to win more, causing more dopamine release, greater reward. So all three do this by three different mechanisms, and that's how these drugs work. Yes?
If caffeine was considered a drug, which it is, right? Caffeine, would that be kind of like the nicotine route?
The caffeine works like the nicotine route.
That's what I was thinking.
Right. And the specifics are a little different. I don't have that up here, but yeah.
I'm sorry, just to reiterate heroin's mechanism, it shuts off the negative and
Yeah.
then thusly
Yeah, remember we're adding these up, right? So it's sort of positive negative. So by decreasing the negative, positive is going to win more than it was before. So because we were at a certain balance, and this was sort of stopping it, if you decrease the negative that's coming in, whatever positive is there.
So almost like the cane though, because you said that whatever was there, it stays longer.
Yeah,
Which
in other words,
makes it win.
like if this were, plus 30, okay? And this were, minus 20, okay? And right now they're both firing, you have sort of a signal of about plus 10. If you shut this off, and this is going the same way, now you've got a signal of plus 30. So that's sort of the idea, because you never have anything on or off. You got them both sort of firing. We'll talk a little more about that. But that's the idea. If you increase this to plus 60, well, that's going to increase it, like nicotine. Or you keep this the same, but you get rid of the negative. When you add it up, this number ends up being more positive. This is going to fire more neurotransmitter release, etc. Yes, sir.
In the same way that a person, I guess, hypothetically, could just take adrenaline, could someone just take dopamine? Not dopamine. Yeah, dopamine.
People do take dopamine that are on Parkinson's, okay, because you're also dopamine is also not just in the reward pathway, but actually controlling muscle movement. The one problem with it is.
But is a thing to just be able to take that one.
Yeah, but it doesn't usually affect this. It's something about sort of the half-life. Because remember, if you're getting it systemically, it has to somehow make it into the synapse here, which is going to be very specific, within the brain, and have an effect. And so I don't think the people that take dopamine, it has much of addictive quality. So, the exact reasons I think are something with that, but I'm not positive. So. Any other questions about that? Again, we're going to see the video, the first 12 minutes, and again, if it's a little confusing, you can't remember everything, it's fine because we're going to go over it all. So just trying to get the feel of it. It's going to talk about how the nerves work, but with these two, the difference between acetylcholine and dopamine. Okay, and in the sheet I have, you can already see kind of what I'm trying to get the difference, how they're controlled, are they fast, are they slow. Then the second half, he's going to do those three drugs: cocaine, nicotine, and THC. And we'll have that. 25-minute video. Then we're going to spend, we might take a break, but then we're going to spend another few minutes filling this in, and the other idea. And then we're going to talk about the lab and do the lab. That's sort of the idea of chapter 6, but again, it's not that important. But we'll see a picture. So is where some cells, the neurotransmitter will bind, immediately causing ions to flow through. Others are going to work through a G protein, which is then going to affect what we call a second messenger that will then, which is cyclic AMP in this case. And that will then cause ion channels to open. If we increase, again, sometimes we do C, big AMP. That's sort of the second messenger in the cell. So ion channels to open, and then the current. So that's sort of, and we're going to put it over there again, that's sort of the two ways that these neurotransmitters work. Some are going to be fast, directly opening up ion channels, boom, ions flow through. Others are going to work through this slower method, not that slow. It goes through a G protein, which causes this messenger to go up or down, and then ion channels will open. Okay? So if we sort of summarize some things with acetylcholine and dopamine then. Okay, and in fact, I don't know, let me look at your sheet because let's see what order I sort of asked things to. Okay. So, so again, we sort of go to the idea of, okay, I just told you the two mechanisms. Okay, one is one, one's the other. Which one is direct ion channels? Okay, it was a very visual video, kind of druggy, like I said, but the visual images usually stick. So direct ion channel. So dopamine uses a G protein, which again, when it binds to its G protein, it increases or decreases cyclic AMP. So when dopamine binds to its G protein, it increases that second messenger cyclic AMP. And that's going to then open up ion channels. So which one's faster? Okay, acetylcholine is faster because it's direct. Okay, the second point that's sort of on there talks about what I talked about. Where's my yellow chart? Talks about what, oh, here it is. I talked about two basic ways that we control levels of neurotransmitter, okay? One was, and the second one was enzymatic degradation, the scissors. Well, that fits one on one on the other. So acetylcholine, which one it is? Which one is it? I don't know what drugs I'm on. Which one is it? Okay, it uses, and we'll talk about this in class, so I don't have to write down the enzyme, but it does use acetylcholinesterase, so it uses, it cuts it up. Enzymatic degradation to control. Whereas dopamine uses the reuptake. So a couple distinguishing features that the first half talked about besides visualizing between these two types of neurotransmitters, acetylcholine and dopamine. One's faster, directly opens up ion channels and uses enzymatic degradation to control. Dopamine uses a slower method, a G protein, the second messenger, cyclic AMP, to then open channels, and also uses the reuptake pump. Okay? And that's a lot of the first half. Okay? Then, again, he explained the cocaine and nicotine. Now for THC, okay, we don't have that kind of written down here. Again, if you weren't hearing before when I was talking, THC doesn't affect the reward pathway as much as this. It's not as addictive, but we have a lot of THC receptors in our, hippocampus, which is involved in memory. And so when we look at THC, is it a direct ion channel or is it a G protein? Okay, so it's going to bind to a G protein, but what is it going to do to cyclic AMP? So that sort of turns off the neuron. Okay, it decreases the likelihood of fire. Again, we're going to make it sort of simple, make it say turn off, and really it's just decreasing the rate of firing, but I can't write that, right? So when it binds to a G protein, it's decreasing the second messenger, which is decreasing the firing, sort of turning off. And if this is a memory neuron, I forgot what I was going to say. No, that's not fun. If it's a memory neuron, like be home by 10 or whatever flew off, that's why short-term memory might be affected. It's turning off neurons involved in memory, maybe learning, we'll see. Not as much infecting here. But what does it do? It turns off neurons. So if it's acting like one of these drugs, and actually increasing reward, which drug would it act like? Okay, THC. Would it fit here, here, or here, based upon what it does? Heroin. Because basically heroin works by turning off a neuron, positive one. We know THC turns off neurons, so the only way it works and fits into the system is if it is like heroin and basically binds to its particular receptors on some of these, turns off some of the negative, and the positive can win. Otherwise it doesn't make sense, because if it were this, it'd be turning this guy off, and that's less reward, and it had nothing to do with the reuptake, pump at all. So that's where THC would fit in, and it does fit in, but it's just, there's not that many, so far, at least by study, there's not that many THC receptors in this pathway, making it a less addictive drug, at least by this way. Yes, ma'am.
So we naturally have THC receptors in our brain.
Yeah.
Do we naturally have THC in our
We have, I forgot the name, but I got it right. I mean, it's on my part book, because it's like, anandamides or something like that. Yeah, we have natural ligands, sort of our natural THC that our body produces.
But how often do those turn off? Like, it's not as often as THC would, right?
Well, I don't know the total numbers, but it's not as, just like a lot of these drugs act like other ones, but they're much more potent. I think our natural ligands, I don't know the exact cycle of it, but they're less potent and less levels. But I don't know, I haven't looked much about, I can't really answer your question, is what I'm saying. I don't know the studies on how much. Yes?
Okay, so there was the one that, so when dopamine turns on the G protein, basically, to release cyclic AMP.
Inside the cell.
Yeah, inside the cell.
Right.
But there are some that are just always on, the THC inhibits.
But what you got to remember, and this is the concept we're trying to get tonight, is with THC, will inhibit when it's on. We're always we always have multiple positive negatives going on. So the idea is, that again, just like these other drugs, that the idea is that if you have more THC now hitting some of these negative neurons, now it's going to result in, the opposite pathway firing more. So we're always we have like, again, they're always firing at certain levels. There's really never off or on. I sort of use that as sort of a easy way of doing it. But you literally are having, hundreds of these sort of inhibitory, excitatory signals, and by modulating, affecting levels of that, results, do you reach threshold or not? Okay? So when the inhibitory, maybe you're winning, you're not reaching threshold and firing, you basically take away some of those inhibitories, and again, now we're going to fire more than before. Did I answer it or make it confuse you more?
You kind of.
Well, what was
No, I think I lost it in between.
you lost the question, or?
Yeah, a little bit, but, well, no, maybe I'll get back to you when I figure it out.
Okay. So, we have a lot of information today too, so we're sort of trying to get it. But, okay, so let me just write these 3 drugs. Okay? Now I've got the drug of choice now. That's not really a good drug actually. It's bad, but it's a good show. So the methamphetamine, actually, I'm putting it over here because it does affect dopamine levels. And it's sort of cool. I mean, I have a picture on a PowerPoint, and I'll load that up for you, but I don't feel like trying to put that up. But basically, it's going to act a little like cocaine, okay? But what it's doing is, let me actually sort of show a picture, okay? And you have that reuptake pump. Okay, and of course we're releasing usually dopamine. Now, cocaine blocked the reuptake, right? What met does, it uses the pump to get inside the cell. And by doing that, it's competing, so that means dopamine doesn't get in and stays out there. But that's not enough. It also then stimulates the release of more dopamine from that terminal. So it's a double whammy. It literally is sort of competing and using the reuptake pump to get in, so dopamine can't. So that's like cocaine in a way, right? It keeps dopamine out because it can't get in. And then the dopamine vesicles that are there, it literally stimulates the release of more dopamine. So more dope, that's how that works. So it's similar to some of these drugs, but it has that little difference of actually not only sort of blocking reuptake, but literally stimulating the release of more. Okay? So that's sort of how MET works. It basically, again, uses the reuptake pump to get in to nerve terminal. Okay, that leaves more dopamine out, but then it also stimulates more dopamine release. That's why it's a pretty potent drug. And it's a lot more than the reward center that all these drugs work. That's why there's all these other effects. Stimulates more dopamine release. Okay? That's the end of our dopamine drugs for today. We've got some serotonin drugs over there and we'll be done. You can see I told you we're going to fill this up with drugs today. So we've done, all of these drugs are affecting dopamine levels. Again, nicotine affects other things in the body as well, but we're sort of talking sort of the addictive thing. So we have a nice little cocktail here. Now we come over here and we're going to deal with another important neurotransmitter, serotonin. Okay? And so basically serotonin is related, again, to a lot of your basic mood, which is normal moods and anxiety, or lessening of it, and sort of the imbalances. So serotonin levels are associated with development of bipolar and depression and various, things like that. And so there's a lot of drugs, and we'll mention in class 2, like I mentioned, selected, in fact, let's put it down here. We have this idea of, where's my yellow truck? I hate these trucks. We have SSRIs, which again are selective serotonin reuptake inhibitors. So you can see serotonin does use the reuptake pump. And what does it sound like in general if we were talking dopamine? We're inhibiting reuptake, that's sort of like a drug,
Keeps it in the synapse.
cocaine, keeps in the synapse. And so, because we don't have enough. Well, guess what? This is pretty cool. And I just looked this stuff up because I didn't know these drugs. Each time I want like new drugs to look up. Okay? I'm putting X because I don't want to write it out and misspell it again. Okay? Okay? Basically, it blocks serotonin reuptake. Okay? Pretty much like SSRIs. Obviously, there must be some difference between Prozac and X, I imagine. Okay? I can't say I haven't been on one of them, maybe, we'll see. Especially if I'm getting older, I might be trying a lot of this, and maybe start getting to this. But X seems a little off my game here, so I'm not doing that. But we're going to put it up there. So it sort of blocks serotonin reuptake. And again, you know, the magnitude and the other body effects sort of make it obviously more of a potent drug than these things. But these things are pretty bad, okay? I mean, in my opinion, if you look at some of those drugs that are out there, and you've seen them on commercials with the sad dogs and the people walking around, then they take the drugs, and the dogs are happy, and the people are just sort of dazed out and smiling. So they really are dirty drugs. They're getting better, okay? But there's a lot of side effects. But you can see, in a way, serotonin is working pretty much like SSRIs, maybe less selective, and that might be it. It's because there's a lot of serotonin receptors. But blocks serotonin reuptake. And so increases serotonin, which again, this basically can maybe overshoot the mark, but changes your mood and stuff like that. Again, I don't know much about ecstasy, I have to admit. Okay, LSD. Well, maybe I know a little more about that, but I won't say. But, long time ago. Well, I'm very old, remember that, okay? So, LSD was cool. I just found this out yesterday looking up. It basically acts like serotonin. It basically binds, it actually has sort of the same shape. It will bind to serotonin receptors and therefore cause the same effect. So LSD basically is a serotonin agonist. Okay? It binds and activates serotonin receptors. Crazy. And that's the last drug we're going to do tonight. I think we're full of drugs. Okay? So, sort of interesting. Okay? At least I think it's interesting. So, that's our drugs for tonight. So, this pathway is important. Again, all this is important. All this is on the test. So, so you want to get this information down and start like going over it as you start going like some weeks. Yes.
So, this is all okay, and like, everything's happy, but then, so when you come down off of these drugs, is there less dopamine in the synapse? Is that why it's so, people are depressed?
Yeah, I mean, basically, because what starts happening is, we haven't talked about, remember we said neurons have a certain plasticity, and part of it has to do with receptors. And so, part of that, I mean, addiction is very complicated and things change, but some things that happen is that basically, as levels of these things go up, you know, we have sort of homeostasis, right? So, the number of receptors now try to counteract by going, a lot of times, down. Now, the trouble is now, when you're off the drug, now you don't have much dopamine, and you don't have many receptors, and so now you're not getting, you're actually worse than you were, right? You're getting less activity, and you're feeling really, really, really bad. And so, but there's other things that happen too. He talked about sometimes you potentiate, and you actually make it where you're more, you know, active to it than you normally would. But again, then you get used to that high activity. When you don't have it, the normal activity, you know, doesn't work. And so, but addiction is even more complicated than that. But that's sort of the simplified answer of that. It starts to make sense. So the amount of receptors sort of try to match the amount of drug, you know, amount of neurotransmitter going in, and then you can get. That's really the part of addiction, right? And then you need more and more and more because you're getting less and less and less of an effect. You see more just to create the same effect. Yeah, I mean, it's not like I'm recommending, you know, high use of these drugs over the next week or so. Okay. Any questions? Okay, so has everyone got like all this? I mean, all pictures and all that, I'm gonna be able to erase part of this board?
Yes.
Okay. So let's finish this up. We're doing sensory lab. So get out your sensory sheet. Again, I'm going to talk a few minutes. Most of the sensory stuff we will talk about chapter 10 next Wednesday. We're going to do